So for those of you watching the video, what we just did is we went and looked at a segment of that Inner Life of the Cell video. This is a really good video, and it's 8 minutes long, or 7:59. It's that version. And we watched from 49 seconds to 2 minutes and 7 seconds. And that part of the video is showing lipid behavior and different membrane fluidity, and that's what we just did. So, okay, next I want to talk about the protein part of the membrane. So we've talked about the lipids and how they act and what they're like. And next we're going to focus on the proteins associated. And the proteins do most of the cool things that are associated with membranes, because they have more variety in their shapes and their functions and the way that they can operate. There are all different types of proteins that can be associated with the membrane. Some of them will be transporting things in and out. Some of them will be letting molecules go through the membrane that can't cross through on their own. Some of them are structural supports. So they go through the membrane and they attach to some kind of fiber on the outside and some kind of fiber on the inside to provide stability. Some of the proteins that are associated with the membrane bind a molecule on one side and send a signal to the other. So they could be detecting a signal from outside and sending a message inside. That's the more common orientation for that process. And some of the proteins that are associated with membranes are enzymes themselves. They're doing an enzymatic process. A lot of the receptors are also doing an enzymatic process as well. So these two groups are sometimes overlap. But a membrane on its own cannot be very effective. It can be a boundary, but it can't do those three big groups of things, like let a cell crawl, let a cell send signals, and let a cell control traffic of molecules. This slide is a vocabulary slide, so it's describing the terms that we use to tell us how stuck in the membrane is that protein. What's holding it there? If we say that a membrane protein is transmembrane, that means the whole polypeptide chain is going through both leaflets. It's going through both leaflets. It's trans across, completely going through the membrane. And most transmembrane proteins, they will have domains. So this is a great way to show what's a domain right here. This would be the extracellular domain right here. Perfect. The extracellular domain, the transmembrane domain, the intracellular domain. So it's all a continuous backbone, but it's got different independently folded regions that are doing different things. This protein has two extracellular domains and probably two intracellular domains and three transmembrane helices. This protein is called a beta barrel, and I have a separate slide about that in a minute. Beta barrel. The next group I want to talk about are D, actually. So this is showing, okay, that's the same protein as was in the green, and it has a binding partner. And these two are non-covalently associated. This is technically with the membrane, but it is with the membrane because it's bound non-covalently to a transmembrane protein. So if I was trying to extract membrane proteins, it would be hard for me to keep this together, and I might not realize, oh, that was a membrane-associated protein. Those can be on the cytosolic side or the extracellular side. This is the same kind of concept. Some proteins are stuck into the membrane, but they are covalently attached to the lipids. So what this red stuff is showing is this protein is covalently attached to lipids, and the lipids are in the membrane. Lipid linked. This one has fewer lipid attachments than this one. This group used to be called membrane associated, and you will still see that term. Membrane associated. So it used to be, you could be a transmembrane protein, or you could be membrane associated. And now, with better research, it's turned out that, oh, membrane associated proteins, they have slightly different kinds of ways that they stay with the membrane. And then this one is relatively new, a monolayer associated protein. And this one is strange, or you're not going to see those very often. Beta baryl, I love those. It gets its own slide in a minute. So if a protein is going to cross through the membrane with part of its backbone, the part that crosses through must be hydrophobic. So now that you have memorized your amino acid classes, you could probably list what some of these amino acids could be that are passing through the membrane. Small nonpolar stuff, not tryptophan, it's too big. And membrane spanning domains are most commonly alpha helix. It's like, let's thread a little bit of nonpolar secondary structure through this membrane, and that's most commonly an alpha helix. What's in here would need to be polar or hydrophilic residues, because there's water here, there's water outside. But if you're looking at a protein sequence and you are looking at amino acids and it's got polar, 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 and then there's kind of short stretch of nonpolar, that's a hint that that might be crossing through a membrane. If it's a big stretch of nonpolar, it might be interior of the whole globular protein. So this is a protein threading through the membrane as a way of holding itself in there. It's a transmembrane domain that holds the protein in the membrane. If the protein is supposed to have the function of letting something cross sides, this is not enough space to do that. So proteins that are going to allow things to cross sides have two major structures. They can form several alpha helices in a ring that creates a pore. And if it's that system, the green parts shown here, those are going to be nonpolar residues because they're facing the membrane. But the channel has to be connected to water here and here. So the interior of the channel would be polar residues. Hopefully not huge polar residues. Small polar residues, so that there's space for things to go in and out. That's one style. And if you read in a paper or something about a protein and it's got 5 is not common, 7 is more common. 7 transmembrane alpha helix forming a channel, it would be that style. If you need a bigger hole through the membrane, those tend to be beta baryls. So the diameter of this passageway is significantly bigger than the diameter of this passageway. So larger larger pores tend to be formed by beta-baryl, and they're named beta-baryl because the whole thing is composed of a cylinder, is the word, a cylinder of beta sheets. What should be the chemical nature of the dark green side of that beta sheet?
Hydrophobic.
That should be hydrophobic because it's within the membrane. The light green side should be hydrophilic because that's lined with water. It's very cool. Love beta barrel. And in beta barrels, the stuff on if this is extracellular, the extracellular domains and the intracellular domains help control what can go through this. It's not just everybody come through. There's selectivity on either side of what can go through. Studying membrane proteins is not easy because they tend to clump together into large clumps because they've got hydrophobic nature. So if you think about, if I'm interested in studying a protein that's in the membrane, it's going to naturally have a lot of hydrophobic nature, and if you try to get it away from the membrane, you can do that, but they're going to clump together. So every hydrophobic part will try to clump with every other hydrophobic part, and you get this insoluble blob of protein. The way that researchers handle this is they extract cellular proteins and they break up membranes with detergent. And some of these detergents we're going to use in lab, SDS particularly. So detergents help solubilize, make soluble membrane proteins. And also, detergents help linearize. I'm writing linearize here. Right up here. Detergents help solubilize membrane proteins, and they help linearize proteins that have a hydrophobic core. And that fact's going to become important next week in lab when we're going to extract protein or look at proteins and determine how long they are. Just go back there. Let me explain that a little bit. So whether a protein is soluble, so if I use the term soluble protein in this class, it's proteins that are not associated with the membrane. They float around. And a lot of the protein in a cell is not soluble because it's literally stuck into or it's associated with membrane. But whether the protein is soluble or not, if I ask how big is this protein, well, it's medium, it's small. The way that we measure how big a protein is we measure either how long it is or what it weighs. The length is much easier to measure. So if we measure the length, we're going to do this by linearizing, stretching out protein, or if it's a membrane protein, stretching out this protein, and we run them through a gel and they migrate based on length. We're going to do that next week. But to do that, we have to get them stretched out in the first place. So detergents can help with the stretching out process and also with making membrane proteins stop clutching onto the membrane. Come with me. Ditch the membrane. I'm not interested in studying membrane. So dumbbell-shaped protein. How can I get it away from the membrane? I can replace the membrane with detergent. And then I can take away the detergent. Or I can leave this in detergent and run it through a gel that way. That's actually what happens.
Is that like the SDS page?
Yeah, SDS page.
Okay.
So sodium dodecyl sulfate, and dodecyl is 12. I hadn't thought about that in a long time. I hadn't thought about the dodecile part of it in a long time, but it's like a replacement lipid almost. It's got a polar end, a non-polar end. So SDS page, it's got detergent, and the page is polyacrylamide gel electrophoresis. That's next week. Let me come back to this in a minute. So, proteins that allow channels through the membrane might be beta-barrel. They may have multiple alpha helices providing a channel. The next class that we're going to look at briefly are structural or anchoring proteins. Anchoring proteins depend on going through both sides and anchoring to something on either side. So strengthening from the outside, strengthening from the inside. So this is a weird example, but last night my son convinced his Wednesday night church teacher that to make him a mask. She had this photocopy of a wolf mask, and he's like, just really needed it. So she went over and copied it. We took it home, and he wanted me to put the string through the holes, and it's just paper, like photocopy paper. So I folded the sides to give some strength. I threaded string through the hole, and then I put tape on either side so it will hold for 1 day instead of 5 minutes. So you need something going through the membrane. You need strengthening structure on either side. And this figure is the equivalent of where I just stopped the video a minute ago. So all of this in the red and blue and green that are connected to each other, everything but these two green tubes. This is part of the cell structural network. And what we're looking at in here, this is the cytosolic face. And like, I know that this is the cytosolic face because I know this stuff is interior to the cell. And so if we are looking at a picture like this, an electron micrograph, we would be inside the cytoplasm looking out toward the cytosolic face of the plasma membrane. And in here, we've got segments of various different proteins. So there's actin. The whole interior face of the cytosolic membrane has an actin cortex that's just near the surface. And it's like crisscrossing scaffolding to provide support. Actin is linked to these spectrin proteins. So if you've ever looked at the inside of a tent, these would be like the tent poles that hold it up. So you've got actin and spectrin complex together. These blue proteins, attachment proteins, these would be transmembrane. Or no, sorry, these would be associated with these red transmembrane. So you can think of this as like the parts that you put together for a tent, and then the plasma membrane is the cloth. But there's got to be something going through. And then out here, these would also have attachments maybe to the outside of the cell. The individual names of these are not important for you to know, but the concept is important. And we come back to this later when we talk about cells crawling or cells moving. They have to somewhat remodel this to move. And down here, these transmembrane proteins, if a cell is going to attach to a surface, if a cell is going to attach itself to a surface or crawl over a surface, there would be a connection through this side of the transmembrane protein to do that. Are any of you using a paper that has like tissue culture stuff? Okay. Tissue culture cells, when they grow on the surface of a plate, they spread out and they attach to the surface. When they're sick or they're damaged, they lose their attachment and they round up. And that kind of assay is a way to determine health of cells. They can't maintain their attachment. Another thing that cells can do with transmembrane proteins is sequester, keep together certain proteins in a certain location. So if these red proteins, I need them to be in a certain location, I can make them stay there by having them associate with transmembrane proteins in that place. I can also use transmembrane proteins to hold neighboring cells together. Those of you who are in histology, if you are in histology, tissues hold together in this way. Like, cell on its own needs to be associated with the neighbor. Transmembrane proteins are involved in holding those together. This situation we're going to look at separately when we talk about transport of transport of glucose in the gut. This is an example of a tight junction. I'll talk about that another time. I'll also use that picture another time. Several of the proteins that are associated with the extracellular face of the membrane will have a lot of sugar chain modifications. So there could be sugar modifications on the lipids themselves. There could be sugar modifications on the proteins that are touching the extracellular space. And all of this together is the carbohydrate layer. It's the carbohydrate layer. And it makes the surface of the cell sugary. So we have a sugary surface, and having a sugary surface gives two important characteristics. So the particular shapes and 3D configurations of the sugar it gives a shape-based identity to the cell. If you have mammalian phys and you talk anything about immune system or recognizing self and non-self, the shapes of the sugars on the surface of our cells help determine what is our cells. Like there's a common set of those for our cells, and it's involved with a major histocompatibility complex, for example. So it provides identity and recognition that, oh yeah, these shapes mean a human cell. The other thing they do is they keep a hydration layer around the cell. So if you've ever, my youngest son, they all love candy, but he did this weird thing where, you know, you give him a Jolly Rancher and he would hold it in his hand and not put the whole thing in his mouth. Like, why would you do that? Just put the whole thing in his mouth. But he insisted on doing that. So, of course, it's like so sticky. The reason that that's sticky is that sugar attracts water and binds it into a layer of hydration or stickiness. So the more sugar, like the sugar-water ratio controls how sticky that is. His was really sticky. So having a lot of sugars on the surface of the cell helps the cell surface maintain hydration as well. Okay. This picture I want to relate to the video. So the overall process in that life of the cell video, inner life of the cell, is showing this process. So there's a white blood cell that is zooming through the bloodstream. And this white blood cell has on its surface particular sugars. These are special sugars. These are not human cell general marker sugars, special sugars. And these sugars can bind to this protein called, or this molecule called lectin. So what the video was showing is what these cells have to do to get a lectin out there to catch a neutrophil. So it was showing from the beginning, there's a signal that comes from outside the cell transmitted to the interior. The cell changes what proteins it's sending out there. It sends lectins out to the surface. Once the lectins are there, they can catch or break, B-R-A-K-E, get this neutrophil to slow down and stay here. Because something is going on where there's an infection down here. So there's an infection down here. The neutrophils are going through the bloodstream. Someone's got to get their attention. Hey, come here. Slow down. And then it rolls. I love that. It was round. It got caught. So when you see the proteins stretching up, it's the lectins binding here. It slows down. What they don't show is it forms some new attachments here and it crawls. It crawls instead of floats. And then it's going to squish through this cell layer. It's amazing. And then it goes in here and it starts literally chemically fighting infection, which is awesome. That's what that video is based on, that whole process.